Welcome to our video on Purified Allergens. I'm Sabine Wünschmann, and I'm the Head of Allergen Manufacturing, Research and Development at Biotechnologies Incorporated. In this video, I would like to discuss the advantages of using Purified Allergens, give you a brief overview of our allergen manufacturing process, talk about pros and cons of natural and recombinant allergens, and give you examples of applications. To start, let's take a look at allergen extracts, which are widely used in research applications. Allergen extracts are essentially protein mixtures. They are highly heterogeneous and have variable allergen content and biological activity. Because extracts are complex mixtures of allergenic and non-allergenic components, they may cause non-specific immune responses. In contrast, purified allergens represent defined proteins with known concentrations. They have verified IgE binding capabilities, low endotoxin, and verified proteolytic activity. Use of purified allergens will increase specificity in biological assays because a single allergen component was used. Also, purified allergens produced under controlled quality conditions have low lot-to-lot variability. Let's take a look at the different steps necessary for purification of allergens. This is an overview of the different steps used for extraction and purification of natural peanut allergens. A peanut extract is prepared using peanut flour as source material. After the extraction, the peanut solids are removed by centrifugation. The extract is filtered and can now be processed for purification of different peanut allergens. Peanut allergens can be purified by affinity chromatography, HPLC, or a combination of both. This slide shows our different natural allergen products. Biotechnologies has a comprehensive line of over 50 different allergen products. Biotechnologies has purified natural Group 1 and Group 2 mite allergens from D. tyrannicinis and D. farinae, which are also available as low-endotoxin or low-tox products. Our low-tox D. tyrannicinis antigen is a potent extract of dust mite culture, specifically formulated for use in most models of allergic inflammation and asthma. Biotechnologies has a growing range of food allergens, including peanut allergens RH1, 2, and 6. Also, we are currently working on purification of soy allergens and fish bivalbumin. Other natural allergens include animal allergens from cat, dog, rat, and mouse, as well as cockroach allergen Belgi2 and ragweed allergen Ambay1. For the development of recombinant allergens, cDNA libraries are constructed from natural allergen sources. cDNA coding for a specific allergen is isolated, cloned into a plasmid, and then inserted into a yeast or bacteria host cell. Recombinant allergens are purified and can subsequently be manufactured under controlled conditions and used for immunobiology and structural biology applications. The cDNA can also be modified to produce individual allergen mutants. One advantage of recombinant allergens is that all batches are derived from the same master cell bank, resulting in consistent quality. Listed here is Biotechnologies' repertoire of recombinant allergen products. As you can see, we have a variety of recombinant mite allergens from 3 different mite species. Also, recombinant cockroach allergens from the German and American cockroach. Our pollen allergens repertoire includes olive, birch, and Timothy grass pollen allergens. In addition, our recombinant animal allergens include Feldy1 and cow dander allergen Basti2, and we also have Alternaria allergen Alte1. This diagram gives a more detailed overview of the downstream processing used for allergen manufacturing at Biotechnologies Incorporated. In this example, natural mite allergens from D. tyrannicinis are purified from mite extract. The source material used for the extract is pharmaceutical-grade mite culture produced under GMP conditions. As you can see, 3 different mite allergen products are removed from the mite extract in sequence by affinity chromatography. All the products are QC checked, which includes SCS page, ELISA, endotoxin, and proteolytic assays. Additional steps may be included to remove endotoxin for our low-tox products, or if QC criteria are not met. Products then pass a final QC check and are subsequently released for sale. Indo Biotechnology's low-tox allergens are designed for any research studies where endotoxin may influence the cellular response. Their endotoxin content is less than 0.03 endotoxin units per microgram of protein. Shown here are 3 different low-tox cat allergens. From left, natural, recombinant, and the deglycosylated recombinant FeLD1. We assess proteolytic activity of allergen products using a fluorescent assay. Hydrolysis of a fluorogenic substrate is measured to confirm cysteine protease activity of DOPI1. A different substrate is used to verify serine protease activity of our detyronecinous serine protease product, and to confirm the absence of serine protease traces in our natural DOPI1 products. A frequently asked question concerns the differences between our natural and recombinant allergens. So in the next slides, I will discuss the pros and cons of natural versus recombinant allergens. For natural allergens, the pros are that it is a natural form of sensitizing allergen, it represents a mixture of the relevant isoforms, and it contains the naturally post-translational modifications. The cons are that natural allergens may contain trace amounts of other allergens, which typically is less than 0.1% and can be monitored using sensitive ELISA assays. There may be some lot-to-lot variability, and there may be limited availability of source material. Pros of recombinant allergens are that they are generally very pure and defined molecules. Production can easily be scaled up to produce 50 to 200 milligram batches. Consistent and controlled quality is achieved with recombinant protein batches originating from the same master clone and being manufactured using optimized procedures. Standardization is easier achieved than with natural products, since recombinant allergens have a defined allergen amino acid sequence, allowing accurate prediction of the molecular weight. Cons of recombinant allergens are certain isoforms have to be selected for those allergens occurring in many isoforms. There may be high developmental costs involved, and post-translational modifications present in the natural counterpart differ or may be absent. Also, the recombinant protein may not fold properly. Once we have a new recombinant allergen developed, it will be validated with the goals to have the best possible correlation with the natural counterpart. A variety of different techniques can be used for validation, which can include IgE binding assays, monoclonal antibody binding, ELISA activity, CD spectra, and biological activity using a histamine release assay. In the following slides, I will show a few examples comparing IgE binding of a few of our recombinant allergens with a natural counterpart. This slide shows a correlation between IgE antibody binding to natural and the recombinant deglycosylated FeLD1. This slide shows a correlation between IgE antibody binding to natural and recombinant DOPI1. And this figure shows a correlation between IgE antibody binding to natural and recombinant DOPI2. Indo Biotechnology's Purified Allergens have been successfully used in many scientific studies. So in the next few slides, I will discuss different applications. Applications for purified allergens include immunodiagnostic assays. In this field, molecular diagnostics or component-resolved diagnostic assays are using purified single allergen components. Purified allergens are used as allergen standards in quantitative assays and as reference materials. Also, purified allergens are used in allergen vaccines. Additional applications include the wide field of basic and applied allergen research. Purified allergens are used for in vitro studies of T-cell function and antigen presentation, animal models of IgE responses in asthma, innate immunity, antibody detection and histamine release assays, as well as structural studies and biological assays. Indo Biotechnology's purified allergens are successfully used by many scientists working on studies using mouse models of asthma, studies on T-cell function and antigen presentation, research on allergen structure and allergen antibody interactions, as well as allergen standardization. For a list of recent publications using our Purified Allergens, please visit our website. Indo Biotechnology's highly purified allergens have been used to solve several crystal structures of dust mite allergens DERP1, DOF1, DERP5, and DERP7, as well as the cockroach allergen BLAGI2 and the mold allergen ALTE1. Shown here is the structure of the mite allergen DOF1, which shows an overall fold characteristic for papain-like cysteine proteases. Further studies include X-ray crystallography of allergen antibody complexes, structural analysis of the group 1 host dust mite allergens DERP1 and DORF1, and complex with an Fab fragment of monoclonal antibody 4C1 revealed the cross-reactive epitope between DORF1 and DORP1 and helped identify residues involved in monoclonal antibody and IgE binding. The image shows the crystal structure of cockroach allergen BLAGI2 in complex with the monoclonal antibody 4C3 Fab fragment and monoclonal antibody 7C11 Fab prime fragment. In conclusion, purified allergens represent defined proteins with confirmed IgE binding, low endotoxin content, and verified functional activity that meet defined QC criteria. Purified allergens are useful for improvement of allergy diagnostics. They may help increase the specificity of research applications by using a single defined allergen component. Purified allergens may be used in allergen vaccines, and they can serve as allergen standards and reference materials. Questions about purified allergens? Please contact me, Dr. Sabina Wünschmann, or Ms. Lisa Wales at Indo Biotechnologies. Thank you for your attention.